Good afternoon, dear students. In the last lecture, we studied how to isolate pure genomic DNA from a bacteria. And the genomic DNA is required for gene cloning or for genetic engineering experiments. Now, today, here we are going to discuss how to separate plasmid DNA from genomic DNA. That means if you want to isolate plasmid DNA from a bacterial cell, how to do that. Okay. So what's a plasmid? It is an extra chromosomal self-replicating, okay, double-stranded DNA that is present. So it is extra chromosomal other than chromosomal DNA. It is self-replicating, okay, and this plasmid DNA in genetic engineering is used as a cloning vehicle. It is used as a vector. So let us discuss how to isolate or how to separate plasmid DNA. Preparation of pure samples of plasmid DNA. Now, purification of a plasmid from a bacterial culture involves the same strategy as the total cell DNA preparation. That means we have studied various steps for isolation of or purification of plasmid DNA. That is growing the cells, harvesting the cells, lysis of the cells, separating the proteins and removing the RNA, and then concentration of DNA. So the basic steps involved are the same. So the culture of cells containing the plasmid is grown in the liquid medium. Cells are harvested. Cell extract is prepared. The cell extract is subjected to deproteinization. RNA is removed and DNA is concentrated by ethanol precipitation method. So after this, the most important step. Now, in your sample, you have two type of DNA, that is genomic DNA and plasmid DNA. Okay, after ethanol precipitation has been done, we have two kinds of DNA in the same solution. What is it? Genomic and plasmid. What do we want? We want the plasmid DNA. So the most important step is plasmid DNA separation of the plasmid DNA from the large amount of chromosomal or genomic DNA of bacteria that is also present. Okay. So here we are going to study how to separate out, how to prepare plasmid DNA. The separation methods of plasmid DNA are based on several physical differences between the genomic DNA and the plasmid DNA. So how to separate the plasmid DNA from the genomic DNA? Based on physical differences. What are those? Size and conformation. Okay, so the most important separation method is based on conformation. Let us discuss in brief about separation of plasmid DNA from genomic DNA on the basis of size. The largest size of plasmid DNA is only 8% of the size of E. coli chromosome. That means plasmids are very small as compared to chromosomal DNA. Okay, and most of the time they are not even 8%, they are much smaller. Hence, we use techniques that can separate small plasmid DNA molecules from the larger genomic DNA molecules. Okay, so we use the techniques based on size. The fractionation on the basis of size is done during the preparation of cell extract. Okay, so here when we are preparing the cell extract, that means the second step, at that time only we carry out the fractionation process. See, if the cells are lysed under carefully controlled condition, instead of using SDS, which will break the DNA also, we don't use strong detergents or strong chemicals. We lyse the cells under carefully controlled condition. Only a minimal amount of chromosomal DNA breakage will occur. That means the lysis of the cell is not done by using strong chemicals. We use mild treatments and lyse the cells under carefully controlled condition such that the chromosomal DNA is not broken and it remains attached to the cytoplasmic membrane. Okay, one end of the chromosomal DNA is always attached to the cytoplasmic membrane. So we do not allow the detachment of DNA from cytoplasmic membrane. Okay, so if the cells are lysed under carefully controlled condition, minimal amount of chromosomal DNA breakage will occur and the resulting DNA fragment will be still much larger than the plasmid and will be removed along with cell debris by centrifugation. Okay, so during cell extract itself we can remove genomic DNA along with cell debris if the genomic DNA is a larger molecule, not a broken one. So how we are going to carry out the process? It is aided, the process of separation of genomic DNA. Okay, removing it along with the cell debris is aided by the fact that bacterial chromosome is physically attached to cell membrane and fragments will certainly sediment along with the debris if the attachment of the DNA is not broken, and hence cell disruption, cell breakage is carried out gently to prevent wholesale breakage of bacterial DNA. Okay. For E. coli and related species, controlled lysis is performed as we'll see in the next slide. Okay. So treatment with EDTA and lysozyme in presence of sucrose prevents the cells from bursting and spheroplasts are formed. Okay. So they have partially lysed cell wall and intact cell membrane. Right. So now here we are going to carry out the lysis under carefully controlled condition. So we have a E. coli cell having plasmid and bacterial chromosome. So chromosomal DNA is a very large molecule attached to the cytoplasmic membrane. Okay, so now we treat the cells with lysozyme and EDTA but in presence of 25% sucrose such that the cell wall is not completely broken. Okay, some fragments of cell wall remain attached but cell membrane is intact. You can see here in this diagram, the cell membrane is intact. Okay. So this is the cell membrane, this is the cell membrane, it is intact. And you can see here, this is cell wall. So some parts of cell wall is present and such a molecule is called as spheroplast. It is called as spheroplast. Right. So the cell membrane is intact and the cell wall is partially disrupted. But you can see here, the DNA, chromosomal DNA is still attached to the cytoplasmic membrane. And now you treat it with a detergent that is Triton X100 and then you get a cell extract. So, but what you get is that the plasmid DNA are present as it is because they're very small in size. But the genomic DNA, okay, because it is not broken completely, most of the genomic DNA is still attached with the cytoplasmic membrane. So when you carry out the centrifugation, because the genomic DNA is attached to the cytoplasmic membrane and it is larger in size, the genomic DNA, larger fragments of genomic DNA will settle down as along with cell debris after centrifugation. And you get a cleared lysate at the top which contains plasmid DNA. Okay. So here we are separating plasmid from genomic DNA on the basis of its size. On the basis of size. So cell lysis is induced by a non-ionic detergent like Triton X100. Okay. And ionic detergents like SDS cause chromosomal breakage. This method causes very little breakage of bacterial DNA. So centrifugation will now leave a cleared lysate containing almost entirely plasmid DNA and removing the genomic DNA along with the cell debris, right? So this is how we separate plasmid from genomic DNA on the basis of size. But the method is not widely used because the cleared lysate invariably retains some of the chromosomal DNA. And sometimes if the plasmid is larger in size, it may sediment along with the cell debris. And hence the separation or fractionation of certain alternative methods are used, or separation of genomic and plasmic DNA on the basis of size is not preferred and some other methods are used. So the next method is separation of plasmid and genomic DNA on the basis of their conformation. Okay, on the basis of their conformation. Now, in addition to size, plasmid and bacterial chromosomal DNA also differ in their conformation. That is, spatial arrangement, overall spatial configuration of the molecule, whether it is linear or circular. Now, most of the plasmid exist as a supercoiled molecule, Why does supercoiling occur? Because the double helix of plasmid DNA is partially unwound during the plasmid replication process by the enzyme topoisomerase. Okay, it undergoes partial unwinding because of topoisomerase, and hence supercoiling takes place. The supercoiled conformation can be maintained only if both the polynucleotide strands are intact. That means if both the strands of plasmid are intact, the supercoiled conformation is maintained. Hence, the more correct technical name is CCC molecule. We call it covalently closed circular DNA. It is covalent, rather than supercoiled, it is covalently closed circular molecule. So if one of the polynucleotide strand is broken, then the double helix will revert to its normal relaxed state, and the plasmid will take alternative conformation called as open circular conformation. Right? So you can see here, this is a supercoiled DNA. So generally plasmid exists as a supercoiled DNA. Only when one of the strand is broken, it acquires a configuration which is called as open circular DNA. So these are the two conformations of DNA. So here separation of plasmid DNA on the basis of conformation occurs when the plasmid is present as a supercoiled DNA. Okay, now supercoiling is important in the preparation of pure plasmid DNA because supercoiling is important in the preparation of pure plasmid DNA because supercoiled molecules can be easily separated from non-supercoiled DNA. Okay, so the plasmid will be a supercoiled DNA and genomic DNA will be non-supercoiled. Okay, DNA. So we now separate the plasmid DNA as a supercoiled molecule from the non-supercoiled DNA. So there are two different methods which are commonly used for separation of DNA from plasmid DNA, and that is alkaline denaturation method and ethidium bromide cesium chloride density gradient centrifugation. Alkaline denaturation method for preparation of plasmid DNA is widely used even today. Okay. So even today in our laboratories, in research labs, alkaline denaturation method is practiced to separate and purify plasmid DNA. Okay. So let us discuss about that method. Now this technique is based on the fact that there is a narrow pH range at which the non-supercoiled DNA is denatured, whereas supercoiled DNA are not. Okay. On what basis we are going to carry out this separation? That there is a narrow pH range in which the non-supercoiled DNA undergoes denaturation. That means genomic DNA will undergo denaturation, whereas supercoiled plasmid DNA will not undergo denaturation. Okay. So you prepare the cell extract, you grow the cell, you harvest the cell, and you prepare the cell extract by cell wall and cell membrane disruption. Now, sodium hydroxide is added to the cell extract or cleared lysate. So that, now what does the cell extract contains? This in the cell extract, the cell debris are separated. That means the broken pieces of cell wall, cell membrane, everything is separated. We have a cleared lysate. What does the lysate contain? RNA, DNA, and proteins. Okay. Now, sodium hydroxide is added to the cleared lysate so that the pH is adjusted to 12 to 12.5. Okay. When NaOH is added to the cell lysate, then hydrogen bonds of the non-supercoiled DNA are broken. Okay. So when you add NaOH, reduce the pH to 12 to 12, increase the pH to 12 to 12.5, then hydrogen bonds of non-supercoiled DNA are broken and the two strands get separated. Okay. So it causes the separation of the two strands of the DNA molecule. Right. Now, if you add acid, if acid is now added to this denatured bacterial DNA, because now the pH has reduced, all the separated DNA fragments will try to re-aggregate. They will try to again get converted into double-stranded DNA. So they will re-aggregate, but they won't get, they will not be properly hybridized, and they will form a tangled mass. They form a tangled mass. This entangled mass is an insoluble network of genomic DNA fragments, and hence it will pellet out after centrifugation, leaving behind pure plasmid DNA in the supernatant. Now here in this step only, the protein and RNA also become insoluble and will be removed during centrifugation. Especially when you carry out the cell lysis by SDS and neutralization is carried out by sodium acetate, you can properly separate the plasmid, because SDS being a strong detergent will break the DNA. Okay. Now phenol extraction method for removing for deproteinization or RNase treatment is not required here. Okay. So now what we do? Alkaline denaturation method or alkaline lysis method for purification of plasmid DNA. What we first do is, we have a cleared lysate which contain linear DNA and supercoiled DNA. You can see in this figure, there are linear DNA and supercoiled DNA. But along with linear and supercoiled DNA, proteins and RNA are also present. Now, to the cell lysate, we add an alkali, that is NaOH, which will increase the pH to 12 to 12.5. When you increase the pH, the linear DNA molecules are broken. That means their hydrogen bonds are broken, and all the double-stranded DNA is converted into single-stranded DNA, whereas the supercoiled plasmid DNA remains as it is. It is not affected in this range. In this range, 12 to 12.5, the supercoiled DNA is not affected. So you can see that they are intact. Now, you add acid. When we add acid, the double-stranded DNA, the single-stranded DNA molecules, genomic DNA molecules, they try to re-aggregate. Okay, they try to regain their double-stranded conformation, and in doing so, they form an entangled mass of linear DNA. This entangled mass is heavier, and hence it will settle down. So this is then subjected to centrifugation. So the entangled mass of genomic DNA will pellet out. Okay, and what we get, a cleared lysate containing supercoiled plasmid DNA. During this process, the RNA as well as proteins are also precipitated, and they are also removed. So in one stroke itself, the genomic DNA, the proteins, the RNA are removed. We don't need any deproteinization or RNase treatment step. We get pure plasmid DNA. And this method of alkaline denaturation is now used also. Even today it is used for separation of pure plasmid DNA. Okay, now right, so here when we add acid, genomic DNA cannot renature. Okay, and plasmids are never, they are never totally broken open, so they can be easily separated. So this is all about preparing purified plasmid DNA, okay, by alkaline denaturation method. The next method is ethidium bromide, cesium chloride, density gradient centrifugation, that we will discuss in the next lecture.